Welcome to the third video under our cognition memory unit. Today we're going to be talking about the biology of memory. We're not going to get too much into really technical details here, but we're going to talk about some of the basics that we know about the biology of memory. Now, if you were to come back to this class or to your notes from this class 20 years from now, I guarantee you that this would probably be one of the portions of the curriculum that has either changed significantly or been fleshed out a great deal or has dramatically improved in terms of our understanding. So with that caveat, let's talk about what we think we know today. Obviously, memory and the storage of memory has to have some sort of biological underpinning. There has to be something going on in the brain, some change that is allowing us to remember that information. For a long period of time, there was a search for what we call an engram. And this engram theory is essentially that you could find the physical trace of a memory in a single neuron. And there was a researcher by the name of Carl Lashley who, with his work with rabbits, trained them to have a specific type of memory. And he kept lesioning out specific regions of the brain. And no matter how many different chunks or how much of the lesions he did, there was still a recollection of that memory. So he decided there was no engram. There's no single engram that you can find and say that is where this particular memory is. And so that led to some different searches and different theories. And our best understanding today is the idea that memories are formation or an activation of a variety of neurons at the same time. And the way that neurons can communicate with each other is through the spaces, the chemicals that are released into the spaces between them called synapses. And what we have found with these neurons is that there is this synaptic change called long-term potentiation. And in essence, what this is happening is that there's a strengthening of the ability of the synapse to one neuron to communicate with another. And the way that this happens is really at the neuron that is receiving the information, it becomes more adept at receiving that information more clearly. It has more receptor sites. It's able to take on more of that neurotransmitter, and therefore that memory becomes stronger. Um, Dr. Webb, or excuse me, Dr. Hebb, a neuroscientist, coined the phrase a few decades ago called that said the neurons that fire together wire together. And that's essentially what's happening. They don't literally come together and wire together, but they strengthen their connections through the synaptic change. And so that's been established in some research, and that's pretty clear. But that being said, it did not kill the engram theory. And there has been some very recent research that suggests that there are certain types of memories that do produce changes at a single neuron level. So I wouldn't put the engram theory to bed just yet, but certainly long-term potentiation is probably the more modern understanding of exactly what's going on in terms of the biology of memory. Now, other biological changes. Stress hormones, specifically cortisol, have some clear impacts on our ability to remember information. And when we get to our stress unit, we'll cover this a little bit more. But essentially, short-term stress helps memory. And if you think about it, if you've ever had a really emotional, stressful situation, you can probably think back and remember that very clearly about what was going on with you. You've experienced a car accident or anything, even something as mundane as you had to get up and speak in front of a large group of people. The short-term stress of that experience activated cortisol, and that cortisol activation, among other things, that cortisol activation created some biological changes in the brain that enhanced your memory of that specific event. The stress response increases glucose to the brain, which is essentially energy. It signals to specific regions of the brain. And I know we have not covered regions of the brain yet. That will be our next unit. But it signals to the amygdala, hey, release signals to the hippocampus, hey, this is important. And in essence, the amygdala is one of our emotional regions of the brain. The hippocampus is the region of the brain that stores explicit, or not stores, but helps process explicit memories. And so the amygdala is saying, hey, hippocampus, this is important. Let's make sure we remember this. And this produces what researchers have called flashbulb memories. Flashbulb memories, if you think of like an old school camera, there's a flash and kind of that picture is produced forever. With flashbulb memories, it's a memory that we experience as highly emotional and highly vivid, and we assume for it to be highly accurate, although we'll talk about a different unit. Actually, later in this unit, that's probably not quite the case. That being said, that short-term stress can also hinder the recall of older memories. And in that way, we have a biological explanation of test anxiety and how test anxiety can negatively affect performance. If you are one who gets super stressed about tests, so there you are with your stress responses going on, you have the increased cortisol levels. What is that is in essence telling your brain is we need to focus on this particular stressful event right now. Forget everything else we've learned right now. We need to focus on this stressful event. And that forget everything else we've heard is hindering your ability to remember whatever you studied for that test. So the key, I guess, to test anxiety is helping to help those who suffer from it kind of mitigate and dampen down their stress response. Long-term stress, for the most part, hinders memory. And the way it does that is because long-term chronic high levels of stress is going to corrode neurons in the hippocampus. Basically, the hippocampal volume is going to be reduced. The hippocampus is going to be able to process new memories less efficiently than it has been before. So, long-term stress, such as living in a war-torn area, living in an abusive household, living through, say, New Orleans after Hurricane Katrina. All of those would be examples of what could produce long-term stress and have negative impact on the hippocampus and therefore negative impact on your memory. And finally, we need to talk about the idea that not all memories are treated the same by your brain. Different types of memories are processed in different regions. And at a basic level, we have implicit versus explicit memories. Implicit memories or procedural memories, these are memories that you don't necessarily have to consciously think of. So, for example, if you're taking notes right now and writing, you're not consciously thinking anymore, how do I make an A, or how do I make, this, whatever it is. That has become an implicit memory. It's become something that you don't even have to consciously think about. And in fact, if you consciously think about it, it may slow you down and make you even make more errors. Implicit memories are processed by the cerebellum, the region of the brain, and the rear, lower region of the brain that is going to help you process that type of procedural memories. In addition, emotional responses are processed by the amygdala and other regions, so that you could lose a conscious remembering of something, you still have an emotional response one way or the other. And in that way, it could be an explanation of kind of deja vu, in that you happen to think that you have this emotional response, you have this emotional feeling, or this eerie feeling about something, but you can't consciously remember anything. And that's partly because conscious memories and emotional memories are processed by a little bit differently by the brain. Typically, we experience that one coherent memory, but sometimes that system doesn't quite work that way. Explicit memories, on the other hand, declarative memories, these conscious memories that we think of. These are things that are like facts that we learned in school, experiences that we've had that we can remember. These are typically processed by the hippocampus. It's important to note that they are not stored by the hippocampus. They're stored in other regions of the brain. But they are processed, and the hippocampus plays a role in helping with that storage, and also helping with the retrieval of that memory, and then pulling it into the coherent whole that we think of as our conscious memory. We're going to take a look at a class case of a gym, um, a kind of classic case study that goes to psychology about a person who, through a surgery at the time where they didn't quite understand what the hippocampus did, and most of his hippocampus removed, and obviously suffered some long-term memory problems with that, suffering what we call interrogate radio amnesia, which is the inability to form any new memories for the rest of your life. So that, that's all right, that wraps it up for tonight. So if you have any questions, as always,